Hello everyone. Before this video gets started, I just wanted to remind everyone that our free online tutoring for the upcoming semester is starting soon. So head over to studystem.org if you need help with chemistry, physics, or biology. Thank you and enjoy the video.
Question 6 of the AP Biology exam. This will be the last question. The question is about the small invertebrate krill species, Thyssanoessa inermis. So it says that they are adapted to cold temperatures, about 4 degrees Celsius of seawater, but recently, due to global warming and other factors, the water temperature has been rising in the area of the krill's habitat, which is making it harder for it to survive since it is adapted to cold 4 degrees Celsius temperature. Now, one effect of higher temperatures is protein misfolding within the cells. But to counter that, krill have several HSP genes that code for heat shock proteins. HSP also stands for heat shock proteins here. So these proteins help prevent protein misfolding or help to refold proteins to their normal shapes. So this is one method that the krill species have used to prevent themselves from dying out at higher temperatures or temperatures higher than 4 degrees Celsius even. The question mentions that an experiment was conducted by scientists on this particular species of krill, and this experiment involved changing the temperature provided to the krill species over and over again. At first, they were given their normal temperature of 4 degrees Celsius, but then they were placed into tanks of 10 degrees Celsius temperature and then back into tanks with 4 degrees Celsius temperature for recovery. And the whole process is analyzing 3 messenger RNAs, RNA 1, 2, and 3, because these 3 messenger RNAs, they are transcribed from certain HSP genes in both the heat shocked krill and the control krill. The control krill in this case is what we call negative control. We talked about negative control in one of the previous questions. So a negative control would be a sample in an experiment that would not be affected by any of the factors that are being used in the experiment. And there is another term that is used besides negative control that is called positive control. And positive control refers to the sample of krill species under the 10 degrees Celsius temperature or under the 4 degrees Celsius temperature under 6 hours of recovery period. This graph shows the 3 different temperatures in which the krill species were placed during the experiment. The first 4 degrees Celsius temperature is when the krill species were in the original seawater, and then the 10 degrees Celsius temperature is when the krill species were taken out of the 4 degrees Celsius bath and placed into the 10 degrees Celsius tank. And then after the 10 degrees Celsius tank, this 4 degrees Celsius temperature refers to the temperature of the water when the krill species were taken out of the 10 degrees Celsius bath for recovery for a 6-hour time period. Now the graph here shows us time on the x-axis and the average messenger RNA concentration on the y-axis. You can see that the average messenger RNA concentration for both the messenger RNA 1 and the messenger RNA 2 is increasing as the temperature increased from 4 degrees Celsius up to 10 degrees Celsius. As for the messenger RNA 1 specifically, its amount or its concentration is increasing even after the 10 degrees Celsius rise in temperature, even after when the krill species were placed in the 4 degrees Celsius seawater for their recovery. whereas there has been no significant change in the messenger RNA-3 concentration even when the krill species were placed in the 10 degrees Celsius path. Part A says, identify the HSP messenger RNA that has the slowest rate of concentration increase in response to heat shock treatment. So using the graph, we know that messenger RNA-3 had the slowest rate of concentration increase in response to heat shock treatment. There was no increase in its concentration even when the krill species were placed in the 10 degrees Celsius path, and it only showed a certain increase in its concentration when the krill species were removed from the bath and placed for recovery in the 4 degrees Celsius temperature. Part B says, describe the trend in the average concentration of messenger RNA-1 throughout the experiment. So for the first 3 hours of the experiment, there wasn't any increase in the concentration of messenger RNA when the krill species were placed in the 4 degrees Celsius path. But when the temperature was increased and the krill species were placed in the 10 degrees Celsius tank, the messenger RNA-1 concentration increased for 3 hours straight. And then when the krill species were taken out of the bath and then placed in the 4 degrees Celsius tank for recovery for a 6-hour long duration, during that period for the first 4 hours of that recovery time, the messenger RNA-1 concentration increased up till there was 25 nanogram per microliter of messenger RNA-1. And after the first 4 hours, the remaining 2 hours showed a decline in the concentration of the messenger RNA-1 concentration. Part C says the scientists hypothesized that the heat shock protein HSP translated from messenger RNA-1 plays a greater role in refolding proteins than does the HSP translated from messenger RNA-2. Use the data to support the hypothesis. So as shown clearly in the graph, when we are comparing messenger RNA-1 with messenger RNA-2, the whole purpose of production of the messenger RNA-1 and messenger RNA-2 shows us that HSP proteins are being made. And HSP proteins, their purpose is the remodeling of the whole structure of the krill species. And the HSP proteins, they would only be required during the recovery period, which was this 6-hour duration when the krill species were removed from the 10 degrees Celsius path and then placed in the 4 degrees Celsius tank. So if the HSP concentration is increasing during the 4 degrees Celsius temperature timing or during the 6-hour recovery period, then that means the messenger RNA that is producing the HSP at that time would play a greater role in refolding proteins, because otherwise there would be no point in having the messenger RNA concentration increase, even though it would be producing HSP. But when the HSP are not being produced during the recovery period, there is no purpose in producing the HSP because they wouldn't be needed anytime other than the recovery period. So what this tells us is that the messenger RNA-1 played a greater role in refolding proteins compared to messenger RNA-2. You can see in the graph that messenger RNA-2 concentration increased up till the 10 degrees Celsius path. But after that, when the recovery period for the krill species started, the messenger RNA-2 concentration decreased when it was supposed to increase so it could help in refolding of the proteins. Instead, messenger RNA-1 concentration increased, which helped increase the amount of the HSP and hence helped remodel or refold the proteins involved in the krill species molecule, in the krill species structure. The last part of question 6 says, messenger RNAs 1 and 2 are transcribed from the same gene, Explain how a cell can produce two different messenger RNAs from the same gene. So a cell can produce two different messenger RNAs from the same gene if it uses two different exons and if it uses two different promoters. Exons are regions of a genome that end up within an messenger RNA, whereas promoters are the regions of the gene from where the transcription of the gene is initiated.